Hello everyone, my name is José Guilherme de Almeida. Thank you for joining me here today as I present to you the work I have developed over the past couple of years on the dynamics and evolution of clonal haematopoiesis. Clonal haematopoiesis is characterized as the accumulation of somatic mutations in haematopoietic stem cells. Over the years, these cells, responsible for the generation of circulating blood cells, accumulate mutations which lead to changes in cellular fitness. If the fitness of a clone, a genetically defined population of cells, increases, it grows at a rate higher than that of other cells. This leads to these clones becoming detectable and to the steady increase in the average clone size, its variant allele frequency, and of the number of detected clones, or mutations, in each individual. Many studies have looked at clonal haematopoiesis cross-sectionally, but few target the evolution and dynamics of these clones. We present here a study that uses longitudinal targeted sequencing of clonal haematopoiesis genes over 13 years and across over 300 individuals. This, together with single-cell phylogenies for 3 individuals, allows us to get a comprehensive view into the dynamics and evolution of clonal haematopoiesis. We model the growth rate of each clone as a linear combination of gene, site, and clone-specific, or unknown cause, effects, and extrapolate trajectories to determine their age at onset. Our model, which assumes clones saturate once they reach their maximum VAF, shows that most clones follow quite steady and monotonic sigmoidal trajectories, with very few outliers being detected. We can also see that gene, rather than site, accurately captures the dynamics of these clones, with SRSF2 P95 age as the one exception. Remarkably, the NMT3A, one of the most prevalent genes in clonal haematopoiesis, grows at an average observed annual speed of 5%, whereas other, much less prevalent genes such as PTPN11 and U2AF1 grow much faster at 20% per year. Additionally, we observe that growth is mostly predicted by the driver, without a lot of variation between individuals. Some exceptions include TAT2 and JAK2, for which the between-individual variation is quite high, implying the role of other factors, such as inflammation. Colonies from individual cells were derived and sequenced for 3 individuals and their phylogenies inferred. The variety observed here is remarkable. On the left, we have a single clone sweeping the entire population. In the center, we have a rich and varied clonal landscape, with multiple clones, including some with no known driver, competing for space. Finally, on the right, we have an individual whose expansions have no known drivers. Focusing on the expansions with known drivers and inferring their phylodynamic trajectory, we see that these estimates agree closely with the ones derived from longitudinal data, both in terms of growth rate and in terms of the inferred age at onset. Here, the dotted line and gray area refer to the trajectory inferred from the longitudinal data, whereas the colored lines and area refer to the phylodynamic trajectory. Next, we investigated clonal deceleration. Our phylogenies and others from collaborators, Emily Mitchell and Peter Campbell, revealed that it is fairly common for growth to saturate without clones reaching their carrying capacity. This led us to look into the possibility that clones decelerate after an earlier and faster growing phase. To quantify this, we fitted a biphasic trend to the phylodynamic trajectory and calculated what the population size would be if the clone had not lost any speed, extrapolating from its earlier growth. We observed that in most cases the observed clade size is much smaller than the expected clade size, thus showing that clones had to slow down. Additionally, we observed that the growth rates inferred from both phylodynamic and longitudinal data show deceleration. Indeed, the late growth is much smaller than the expected growth for the phylodynamic data, while the observed growth is much smaller than the growth that would lead to an age at onset estimate that does not exceed lifetime in longitudinal data. We also hypothesized that clones characterized by mutations that confer small fitness advantages, the NMT3A, BRCC3, and TP53, are the most affected by deceleration, while the ones with mutations conferring large fitness advantages, SRSF2, P95H, U2AF1, and PTPN11, show little evidence for this. Finally, we calculate the expected range for the ages at onset. Particularly, we observe a consistent accumulation of clones through life for all drivers except SRSF2 P95H and U2AF1, both of which confer large fitness advantages and are largely absent from myeloid malignancies at young ages. As concluding remarks, we can now make a few statements about the evolutionary dynamics of clonal haematopoiesis. Time series data from over 300 individuals reveal steady and consistent trajectories of expansive mutant clones. Different driver gene mutations exhibit different annual growth rates, with the lowest occurring for the NMT3A at 5% and the highest for U2AF1 at 20%. Phylogenies from 3 individuals confirm constant growth rates over 3 to 5 decades and reveal concurring clonal expansions without known drivers. Some clones slow down during their development, and this can be caused by competition. The onset of clonal growth is estimated to occur within the first 3 decades of life, with exceptions such as clones characterized by U2AF1 and SRSF2 P95H. Finally, I would like to thank my supervisor Moritz Gerstung, my collaborators on this project, especially Margit Fabre and George Vassiliou, and Emily Mitchell and Peter Kempel. I would also like to thank the rest of the Gerstung group and you for your attention.